And there's some really famous ones in Saccharomyces cerevisiae that are called killer viruses.
Great band name. Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am Kim Klonowski.
And I am David Peterson, your hosts for today's episode. Can jumping genes help us fight a deadly fungus? Today's guest is using them to identify the weak points in a pathogen's armor.
Our guest today is Dr. Blake Billmyre, Assistant Professor in the Departments of Pharmaceutical and Biomedical Sciences and Infectious Diseases. Blake, welcome to the podcast.
Thank you for having me.
So our standard first question now is, how did you come to science? What directed you to this career?
Yeah, I really got there more by default than anything else. I started as a biology major at the University of Maryland and discovered that I really liked being in the lab. And so I worked in a bunch of different labs. And eventually, after a short stint as a postbac studying mice and cancer immunology at the NIH, I moved to grad school at Duke University, where I ended up deciding really that I wanted to work on microbes. I didn't have anything else in mind other than it needed to be single cell. My final rotation in grad school ended up in Dr. Joe Heitman's lab there, where I was sort of tricked by the incubators on my first day. I opened them up, and it had this amazing bread smell from the yeast growing in the incubators there. And little did I know there was only one person there that was still working on baker's yeast. And everyone else had switched over to pathogens, primarily Cryptococcus. And it turns out, and I've worked on Cryptococcus the rest of my career, it smells absolutely terrible in an incubator. But I stuck with it anyway. So
is that a general feature of pathogenic fungi? They smell bad?
I think it's a good question. I've never sniffed a plate of several of the things. So some of them it's probably a bad idea.
Well, the first thing that comes to mind to me is toenail fungus, but that's a whole nother level.
That one does smell bad. I can attest to that. We've grown some of that before.
Oh no. So what's the fundamental question that drives your research?
So I'm really interested in diversity, evolution. My lab focuses on understanding evolution at a few different timescales. So we're really interested in the evolution of virulence, evolution of pathogenicity. You know, how do we get things that become virulent to hosts, but also thinking about evolution, during infections, for example. So how do we evolve resistance to antifungal drugs? How do things adapt to the host they're growing in? So generally focused on those types of questions in my group.
So since we're talking about diversity, one thing that interests me is the fact that there's pathogenic and non-pathogenic species of Cryptococcus, and what differentiates them?
Yeah, so it's something we think about broadly for fungi. Is fungi are everywhere. They're fairly ubiquitous, and very few of them are pathogenic to humans and mammals in general. And so one of the barriers to this that's been proposed by Arturo Casanval and others is the sort of thermotolerance barrier, that very few fungi are capable of growing at human body temperature, for example. And we don't know yet whether this is something that's universally true, whether this really explains all the virulence differences, but at least in Cryptococcus, it's suggestive. The pathogens within the Cryptococcus genus are split from the non-pathogens over a period of about 100 million years ago. And those pathogens are all capable of growing at human body temperature, while the non-pathogens are not. So this is actually one of the questions my lab's very interested in exploring is understanding how thermotolerance evolves over time and what sort of genetic changes lead to that.
Some veterinary animals have higher standard body temperatures. Do they have less fungus?
So it's interesting. So one of the pathogens that's a really prolific pathogen of birds is actually Aspergillus. And Aspergillus notoriously has an incredibly high growth temperature. So it typically grows in compost piles, for example, which often vastly exceed human body temperature. So it's certainly not a hard and fast rule that fungi don't like the heat, but the ones that do often show up in animals as well.
So we talked about Cryptococcus being all around us, but we all don't have to be necessarily worried about Cryptococcus, correct? We don't want to scare our listeners.
No. So in fact, many, as I said a little bit earlier, many of the fungal pathogens are fairly ubiquitous, right? So we encounter these more or less all the time, but the difference between a person getting infected or not typically has to do with your immune system and how you react to that pathogen. So many of the fungal pathogens that do cause that disease in people who are immunocompromised for one reason or another. In the case of Cryptococcus, this is typically in conjunction with the HIV and AIDS epidemic. So the vast majority of Cryptococcus cases occur in sub-Saharan Africa in conjunction with poorly managed HIV.
Was Cryptococcus one of the first immunocompromised diseases they were seeing in HIV patients?
It was, and Cryptococcus is particularly common in patients with advanced HIV, and so it's actually one of the AIDS-defining illnesses, meaning that patients who are diagnosed with HIV progress to AIDS when they're diagnosed with one of several different diseases that are prolific pathogens of immunocompromised individuals. And so Cryptococcus is the AIDS-defining illness for the majority of AIDS patients.
So one of the tools you use to study Cryptococcus are transposons.
Yeah.
So tell us a bit about what a transposon is.
Yeah, so a transposon is a selfish DNA element that exists more or less to propagate itself. It often does so to no benefit of the host or even at the detriment of the host. So they're made famous by Barbara McClintock originally, who identified them as jumping genes in maize. So the sort of difference in kernel color in maize is often determined by movements of these transposons. So the type of transposon that we typically work with are transposons that basically cut themselves out from the genome and they reinsert them in another location, sort of a cut and paste transposon.
So are the cut and paste transposons, is that a random event? It's literally almost taking a chance?
Effectively. So they have differences in bias and where they like to go. So some transposons do have sequences they like to insert into or locations in the genome they like to insert into. Others have very little. In our case, we like to work with something with as little bias as we could. We're taking these transposons to use them as tools rather than directly studying their biology. But there's lots of variation in the types of transposons between different organisms.
So did you have to do some work or modify the transposon to whose natural home is maize to work in Cryptococcus, or does it not care? It's just DNA's DNA.
It cares surprisingly little. So we tried a few different transposons in the process of getting this to work. We actually started with the transposon that came from houseflies because it had worked pretty well in other fungi prior to setting the system up. And for whatever reason, we never totally understood why. It didn't work nearly as well in Cryptococcus as it had in some of the others. But the maize transposon pretty much worked right out of the box. For whatever reason, it moves around really well in the Cryptococcus genome. It does have some of these biases in where it likes to go. So there's parts of the genome it likes to insert into more than others. It actually likes to avoid genes. But in a genome the size of Cryptococcus, we can sort of view these transposons as sort of a sledgehammer. We can just really hit it hard and not worry so much about these biases.
So can you kind of walk us through a little bit more about the experiment? So how many genes do you target at one time? How do you determine that a gene is interrupted?
Yeah, so what we do is we make massive mutant libraries where we mobilize a transposon in a pool of cells with the hope that every single mobilization event moves the transposon to a new random location in the genome. So we do this in a massive pool in one culture where typically we end up with more than 1 million transposon movement events in a genome that's only about 19 million bases. So we end up with really, really high density coverage where every single cell in that pool has a single random transposon in one random location in the genome. And then we map those transposon locations using targeted next-gen sequencing. So we just sequence the boundary between those transposons in the genome, and we can make this massive map of where all the transposons are, where they're not, and we can infer a lot of things about phenotype from that.
So each cell has just a single transposon copy, and then you activate it in some way to cause it to move?
Yeah, so we start with a pool of cells where they're a single copy in one location, and then we have a transposase at another location in the genome that we can trigger by growing these on galactose, so a specific kind of sugar. We move them all in a pool. We take away the sugar so that they no longer move anymore, and then we work with our sort of static set of transposon inserts from that point on.
And then I guess it comes down to having a good assay to look for whatever feature or characteristic.
Exactly, yeah. So I really like drug resistance for a lot of reasons. The first of all, it's important, but the more cynical reason is it's really easy. It's the easiest selection you can do to basically take cells and put them on something that should kill them and see things that don't die, right? So we can do these sorts of assays for drug resistance in bulk, where we basically ask across the entire genome, are there genes that don't tolerate transposon insertions once we add this drug in? Or are there cells that have transposon insertions that grow even better in the context of that drug? And so we can basically ask for things we could design synergistic drugs to target, or we could look for things that would predict what might be happening in a patient that's being treated with that drug.
So you could look for gain or loss of a function.
Exactly. We can do the assays both ways.
Can we just break it down a little bit for a listener who's listening? So the idea is that you have something that a drug that you're treating with, and you want to know whether or not, I'm going to put it in your words.
Sure.
You're going to do a better job.
Yeah. So one of the things that was really fascinating to me actually at the tail end of my PhD is we sort of stumbled into drug resistance for a totally different reason. We were actually we're working on an outbreak of Cryptococcus gattii in the Pacific Northwest of the United States, and essentially doing genomic epidemiology. And one of the things we stumbled on in that process was a subset of the strains had a mutation that caused them to mutate much more rapidly than others.
Okay.
And we thought, well, maybe these will make them acquire drug resistance more quickly. And in the process of doing these experiments, we started taking clinical antifungal drugs and growing Cryptococcus on them, showing they develop resistance more quickly if they had one of these hypermutator alleles. In the process of doing this, we realized that for several of the antifungals, we actually didn't know how they worked. We were treating patients with these things, and nobody had ever actually done any of the resistance experiments in Cryptococcus. We were basing all of our knowledge for one of these drugs, in particular flucytosine, on really classic work that had been done in budding yeast decades before. And that budding yeast is 600 million years of evolution diverged from Cryptococcus. So in the process of doing these experiments, we started to find new resistance mechanisms, including pathways that don't exist in budding yeast. So we're really interested in using these transposon approaches to basically ask the question of whether there are mechanisms of resistance that exist in Cryptococcus that we don't already know about. And can this help us to understand what's happening when we treat patients with those drugs, and to help us understand how we might be able to design better targeted therapies in the long run.
Okay, right. So almost essentially kind of pathways you don't want to go down because you know it's going to lead to drug resistance in a very likely way.
Exactly. So this particular drug, flucytosine, which is probably one of my favorites, is a terrible drug. It works really well for treatment if you treat with something else simultaneously. It works really well as a combination therapy. But if you were to just treat a patient with flucytosine, they develop resistance incredibly rapidly. We just swap this on a plate, it develops resistance almost instantly. But it works really, really well as a combination therapy. And so understanding how we could maybe handle some of these resistance mechanisms and potentially design other drugs that might work really well with this first one, I think would be really powerful for treatment.
Yeah, and I think that's important to know for two reasons. The first being that some of these treatments are not nice.
No.
Right? So if you can avoid giving someone a horrible treatment that's not going to work, right? Let's do that.
I think I've heard amphotericin, which is a very common antifungal, referred to as amphoterrible.
Exactly. Yeah. So amphotericin, our sort of frontline treatment for treating Cryptococcus, this is typically amphotericin in combination with flucytosine. And flucytosine, on face value, should probably not be a very well tolerated drug either. It's actually, its mechanism of action is as a prodrug that gets converted into a second drug called 5-FU, which people more commonly know as a cancer chemotherapeutic.
Mm-hmm. Mm-hmm.
And so the only real range of efficacy for us in treating fungi is that human cells don't have the transporter to take up 5-FC, and fungi do. And so we start to get toxicity from it when your microbiota starts converting 5-FC or flucytosine into 5-FU, and you're treating your patients with chemotherapy at that point.
So why is it so hard to develop good antifungal drugs?
Yeah, there's a few reasons, and one of them is the fungi are relatively closely related to humans. We're used to thinking about drug development, antimicrobial drug development, at least in the context of either bacteria or parasites, where there's quite a bit more evolutionary distance. But fungi are actually a pretty close sister group to the animals, and so there's relatively little space in terms of drug design that doesn't overlap with mammalian biology as well. It's one of the reasons that cerevisiae or budding yeast has been a really great model for understanding human biology for a long time, because most of the basic biology that you can figure out in budding yeast turns out to be true in humans too. But that's a double-edged sword when it comes to treating these infections. The other part of it is that they have often relatively large population sizes, and they really like to express drug efflux pumps. So it's relatively easy to get resistance during an infection relative to some of the parasites you might worry about a little more, even more closely related to humans, don't have the sort of population sizes that a fungus does during infections.
Yeah, so to get back to my point where I was 1 and 2, 2 was the combination, which you're kind of talking about now, which I think is interesting, because I think a lot of times we also think when you're thinking about drug discovery, you're thinking about things as a single drug or a single resistance, but most of the time these people are not treated with just one thing, and sometimes they have multiple things happening that aren't just maybe fungal. So there's a lot of things to consider. You brought up something that I didn't think to talk about, but now I'm curious about it, is the microbiome. Yeah. So how does your field deal with diversity amongst the microbiome?
Yeah, so we don't do quite as much in Cryptococcus because there's not quite as much impact for the microbiota, at least that we appreciate. There's not quite as much in, say, the lungs and the brain. Certainly there's some microbiota in the lungs, and I know there's some evidence the microbiota in HIV patients is altered within the lungs, but it's not something we've historically dealt with. For some of the other fungal pathogens, and there's maybe a suggestion for a future guest, there's a lot of evidence. Candida albicans, which is one of the sort of big 3 fungal pathogens, is already part of our microbiota. Mm-hmm. And so a lot of the infections with Candida appear to be via disturbances of the microbiota and failure of your immune system to maintain its sort of tuning of the immune system. So there's some really, really nice work from Kyla Ost, who happened to be a grad school classmate of mine who now has a lab at University of Colorado, Anschutz, showing that the immune system essentially tunes Candida to prevent it from growing in a pathogenic form, and that there's some relationships between chronic GI issues and Candida albicans as well, where she's shown some evidence you can basically vaccinate against Candida to prevent colitis symptoms. So,
oh, interesting. Yeah, because we don't think about fungi. Well, I say we, meaning maybe me, think about fungi when we're talking about the microbiome, but it is part of it.
I think that's generally true. When someone mentions the microbiome, you mm-hmm. think of, well, yogurt and mm-hmm. and then bacteria.
Bacteria, yeah.
But yeah,
it's viral too in some ways. It can be.
Yeah, exactly. I think it's for a long time been partially affected by how people have collected the data for the microbiome. So relatively rarely are they actually doing the metagenomic sequences that you need to measure the fungal components. But we do appreciate that Candida is incredibly common in the GI tract. One of the dominant parts of our skin microbiota is actually a fungus called Malassezia that's responsible for things like dandruff, for example. So people who have inappropriate allergic reactions to antigens produced by Malassezia have dandruff reactions to it.
Fascinating.
I did not know that.
Yeah, so the Head and Shoulders
I was just going to ask, what is in Head and Shoulders?
Yeah, Head and Shoulders shampoo is essentially an antifungal that's trying to clear out Malassezia.
Interesting.
Do you have to make sure people in your lab don't shampoo with that? Or
I actually do have a funny story about that.
Oh, no.
So I want to jump back to just after you use your transposon screen and you've identified a line that has a disruption in a gene that either causes resistance or removes it. What's your next step and what other tools are you using?
Yeah, so I've often referred to my lab as sort of a damp lab. So labs are often classified as this either wet experimental biology or dry computational. We sort of straddle the line between the two. We do a little bit of both. So the genomics approaches we use, like TNSeq, sort of they straddle the line, but they maybe even fit a little more into that sort of dry lab side. We tend to go back in afterwards with very classic genetic approaches. We can make targeted mutants deleting these genes and then explore what the phenotypes of those are. And so we're just digging into some of these interesting phenotypes we found from our original screens now, including enzymes that we don't have any idea yet why they're playing a role in drug resistance for the most part. But some really cool phenotypes we found that were unexpected. So, for example, the screen we've done so far with one of our really common antifungals, fluconazole, we found evidence that perturbing mitochondrial function actually makes you resistant to this antifungal. So something that sort of uniformly makes the cells grow more slowly and more poorly is conversely making them much more resistant to drug. And so we're exploring some models right now, including potential stress responses that might be upregulating drug efflux pumps and things like that. But we're not sure yet. It's one of the exciting things right now is being able to dig into these things. And
biology is cool.
Yeah,
it is really cool. Yeah. So are you finding overlap in pathways? I mean, you have the mitochondria, so are you finding many things that kind of affect that same pathway?
Yeah, so we have found mutants that hit almost anything in mitochondrial metabolism. So mess up the entire electron transport chain, any part of it, and that'll make you resistant to drug. And what we're digging into now, I will admit in part because of reviewers, is whether or not these are generalizable responses. So does perturbing the mitochondria make you resistant to any drug, or does it make you resistant just to a subset of these antifungals? And so we're excited. It's a good question, and so it's something we're excited to go after now.
Yeah, sometimes reviewers actually do a good job.
Yeah.
So you mentioned, I think, that you've got 19 million bases in the genome. How many genes does it have? And for reference, humans have 22 to 25,000. I don't know what the current count is.
That sounds about right to me.
Yeah.
Cryptococcus is a little more simple, so it's about 7,000 genes. One of the other projects we're working on right now is actually trying to understand gene count, not just at sort of an individual strain level, but thinking about genes on a species level as well. One of the graduate students in my lab has done some really exciting work looking at what we call a pangenome. We're basically assembling what's the entire genome that's segregating throughout a species. And we found several thousand additional genes that are found in a subset of strains, but not all of them. This is actually one of the other things we're interested in using this transposon approach to understand is what are the function of these genes that are sort of optional in the Cryptococcus genome, and what could they be doing in terms of virulence or just in general growth for Cryptococcus as well.
So knowing what you know about evolution of these species, do you think that a targeted approach is maybe going to be the first good antifungal that we're going to see out there? Or do you think we're going to be able to, not we, you, will be able to identify that's something more broadly applicable to many different fungi?
Yeah, it's sort of this is one of the times I have to put on my cynic hat a little bit, unfortunately, that one of the problems we have for antifungal drug development is that it's not very profitable for the most part, right? So many of the cases for Cryptococcus specifically are in sub-Saharan Africa, where there's not a lot of money to pay for drugs. And so pharma doesn't have an enormous incentive to spend $1 billion to get things through a clinical trial when they won't get the money back. So what we've seen in recent years tends to be more broad-scale antifungals. And one of the most recently approved drugs, a drug called Brexafungerp, which is a wonderful name, was actually approved off of clinical trials for vulvovaginal candidiasis, because that's a recurring problem that you see in countries in the first world that have lots and lots of money to pay for drugs. So it's actually being used off-label for invasive fungal infections where we probably had a greater need for a new therapy, but that argument tends to be the one that pushes things through. So for the most part, I think where we're going to see progress is therapies that are broad enough that we can use them for multiple diseases, multiple body sites, and potentially in agriculture as well.
Well, and hopefully everyone wins.
Hopefully.
So let me ask a question about another aspect of your research, because you found a DNA binding protein which is derived from a virus, which I find interesting because transposons are derived from viruses, right? So you used an ancient virus to find part of a virus, and that has a role in Cryptococcus by binding to chromosomes.
Yeah, this is one of the other ones that we're still very early stages on, but we find really exciting. When I was a postdoc, I took a brief detour away from pathogens and worked on one of the model yeasts, which is a model fission yeast called Schizosaccharomyces pombe. And pombe has this virally acquired gene that binds to the telomeres, so the ends of the chromosomes, and it tethers them to the internuclear membrane. And this virally acquired gene isn't even present in an awful lot of fungi, but the other ones where it is, we don't really know very much about what it does. The one thing we know is it's not particularly important even for the nuclear membrane tethering. It's totally dispensable for viability in Pombi. But our initial TN-seq assays found that this virally acquired gene was totally essential in Cryptococcus. And so we're now sort of left with questioning whether or not it's even doing the same thing. Is it tethering the chromosomes to the nuclear envelope? Is that somehow important in Cryptococcus when it isn't really that important in Pombi? Or is it doing something totally different? Has it acquired this thing from a virus and used it for a different purpose in Cryptococcus than it's used it for in Pombi? So we're really interested in finding out answers to those questions. Those are one of the things I've been shouting this project at every grad student I come across in the lab to try and get somebody as excited about it as I am, and hopefully we'll find an answer to that at some point in the future.
I think just thinking about a virus that infects a fungi in and of itself is fascinating.
Yeah, I very briefly, as a graduate student, thought I was going to be a virologist. When I started my PhD, I was going to work on mycoviruses, so viruses that infect fungi. And there's some really famous ones in Saccharomyces cerevisiae that are called killer viruses.
Great band name.
It's a bad name, yeah. So killer viruses are actually beneficial to their host. So they're a cytoplasmically encoded virus that produces a toxin and an antitoxin, and it pumps the toxin out of the cells and it kills neighboring yeasts that don't have the virus. So it selects for itself.
That is diabolical.
It's diabolical, yeah.
Another horror movie that comes out of the podcast.
Yeah, so they select for the virus to essentially propagate through the population, but they do it while benefiting their host by killing non-related competitors. And so when I started grad school, there's this very cool correlation between a set of genes that were present and they're involved in viral defense called RNAi. And so budding yeasts that had these killer viruses had lost RNAi, which if you reintroduced will actually silence these viruses and prevent them from killing their neighbors, their competing neighbors. And the subspecies of Cryptococcus that was causing this outbreak in the Pacific Northwest of the United States had also lost RNAi. And so we spent a lot of time trying to find these killer viruses in Cryptococcus as well, only to years later, after completely failing on this project, discover that they did in fact also exist in Malassezia, the fungus that's growing all over our skin, that has also lost RNAi. And if we took the isolation protocols that we used to grow Cryptococcus from the environment and grew Malassezia in those conditions, it cures the virus. And so we had screened an entire culture collection that would have been cured of whatever virus we were looking for from the start.
Oh, no. Oh, great story, though.
That is a great story.
Our guest today is Dr. Blake Billmyre, Assistant Professor in the Departments of Pharmaceutical and Biomedical Sciences and Infectious Diseases. Blake, thanks for being with us.
It was a pleasure.
Links to additional information about Dr. Billmyre's research can be found on our Instagram account, which is at PPPPodcastUGA.
As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched last season. Please rate the podcast. I'm thinking 5 stars. And if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases at the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Thanks to the New Media Institute at Grady for use of their Studio Not Found podcast facilities, and a special thanks to our production assistant, Sid Wigand, for research, editing, scheduling, and keeping us on track.